I really appreciate you guys having me. I love to talk about bees and the research I'm doing here in North Carolina. And so I wanted to tell you a little bit today about fiction, fact, and pheromones related to honeybee health. So let's see. Um, I'll just start with an outline of what I'm going to tell you about. So I wanted to start with some honeybee importance, honeybee health information. I'll talk to you about my main topic of study, which is hygiene and brood signaling, and then a new tool that we're working on developing to combat Varroa mites. A little bit of information about foundation wires and an experiment I did a few years back that I think you'll find pretty interesting. Kind of the overall importance of the work, where we plan to go from here, and then I hear you guys are good question askers, so um, I'll I'll definitely stick around for questions if you guys have any. So when I started working with bees and beekeeping 10, 12 years ago, I think that the public sentiment towards bees looked something like this. That's kind of how I felt about bees as a new bee coming in and learning about bees. But I think that actually the increased media and attention for pollinators has brought people closer to the reality of bees. Not that we don't have a hot hive every once in a while, but the reality of bees is that they really are pretty gentle creatures and amazing creatures. Um, they're fascinating, and I think everybody finds honeybees fascinating for different reasons. You guys are all experienced beekeepers, so you know that once you kind of get into beekeeping, you just get sucked in. They're so interesting. And for me, one of the reasons I find honeybees so fascinating is because of their complex social structure. So you know that they have three castes. They have the drone, the queen, and the workers. The drone, of course, mates with queens from other hives. The queens lay all the eggs, and then the workers complete all of the other hive tasks. And one of the really interesting things with the workers is that those tasks can change as the workers age. So, of course, the younger bees are doing brood tending in the nest. They kind of tend to do all those things that are inside the colony that are less dangerous. And then as they age and are actually less valuable members of the colony in some ways because they're older, maybe closer to the end of their lifespan, then they go out and they do those more dangerous jobs of foraging for nectar and pollen. And so, as a biologist, honeybees are really important. This kind of plasticity in their behavior makes them a really important biological model for studying behavior, but they're also really important for studying things like communication, studying nutrition, aging, and genetics. And one of the reasons for that is they display something called phenotypic plasticity. So they can go from one genotype to two different phenotypes. And of course, what I mean by that is, or an example at least of that, is with diet. So if you have a fertilized egg that is laid by the queen, the orientation of the cell and the diet that larvae is fed will determine who it becomes. And you just see this really cool divergence in things like lifespan. So, of course, if that larvae is fed royal jelly, it develops into a queen. Well, I know this isn't the, the lifespan we tend to see with queens anymore, but typically, pre-Varroa, this is the lifespan of queens is 2 to 5 years. And so biologically, it's really fascinating that just by changing that cell orientation and that diet, you get a change in a lifespan, depending on season, of 3 to 16 weeks to 2 to 5 years. So honeybees are really cool to study as a biological model for things like aging, nutrition, and genetics. Another reason we all know honeybees are important is as a crop pollinator. So they contribute billions of dollars every year to the US in terms of crop yields. The estimate globally is between about 235 and 577 billion dollars every year. We're all familiar with migratory routes of commercial beekeepers, and this is really a huge importance of honeybees in terms of food security. And of course, our diets. I know there's one chef here, Sue, but our diets are much more interesting. Our plates more beautiful with bees than without. But how about the environment? So there was a paper that came out in 2017 that I found really interesting, and it showed that of tens of thousands of pollinator species, so there's so many thousands of pollinator species, um, honeybees account for 13% of floral visits. So just being one species in those tens of thousands, they represent such a huge number of visits to flowers in natural ecosystems. So this is not crops, but just in natural ecosystems. But the same paper also pointed out that honeybees actually failed to visit nearly half of all animal pollinated plant species. So half of the species that need an animal to visit for pollination, honeybees will never visit. So what that tells us is that native bees really are critical to natural ecosystems. But we know, and as you can see in this picture, that honeybees and native bees share resources. And because they share resources, they also share pesticide exposure and they share parasites. And so honeybees are also very important for the environment, not just because of the natural ecosystem pollination that they do, but also I think because we don't manage native bees that often. And so, in a lot of ways, honeybees can act as a canary in the coal mine. They can really tell us if there's a problem. So, because they're a managed species, in some ways like a livestock, if we start seeing issues with honeybees, then we can infer from that that there may be issues with native pollinator health as well and take action. And controlling honeybee parasites because of those shared resources may actually benefit um, native bee health in addition to honeybee health. So, bringing us to honeybee health, is there a bee apocalypse? So, we were talking about it a little bit before. Starting in about 2007, 2008, we started seeing all the news networks, media giving lots of attention to honeybee health. So there was the vanishing bees threatening U.S. crops. Be afraid. Time magazine put out a world without bees. Um, but the big message from the media was that colonies are in collapse. And everybody started talking about colony collapse and CCD, which I think is great because it got a lot of attention focused in on honeybees. But actually, colony collapse disorder is a very specific set of symptoms, and a lot of people kind of think of it as a more general thing. But colony collapse disorder is a specific set of symptoms where you find few adults in the hive, but there aren't a lot of dead adults outside the hive. They're just kind of not there. But the queen, brood, honey, and pollen are all still present in the hive. So it's a really a specific thing. And if we look at the millions of hives in the U.S. over a longer period of time, you can see that there was a decline in the number of hives in the U.S. long before CCD was first reported. So to understand kind of what's going on here, we need a little bit of a history lesson. So the most colonies that the U.S. ever had was during World War II. And the reason for that is one, wax was really important for the waterproofing of military equipment. It was used for bullet lubrication. But also, there was a huge demand for use of honey by the public because sugar was kind of being saved for Allied forces. So when the war ended in 1945, that demand decreased, and the number of colonies in the U.S. Started to decline. There just wasn't as big a demand for honey. Um, you can see around the 70s, we kind of hit like a stable number of colonies for maybe a decade or two. And then in the late 80s, the parasitic Varroa mite was introduced, and it kind of set off this secondary decline in colony number. Um, and actually, since about 2008, we've started to see the number of honeybee colonies increasing again. And like I said, I think that's mostly has to do with increased awareness, increased media attention, and maybe also some increased funding associated with pollinator health and education. But just because the number of honeybee colonies is increasing doesn't mean we're in good shape. So honeybee health is still, I would say, on the decline. And I'm sure a lot of you are familiar with the Bee Informed Partnership, but it's a really neat program, set of surveys for keeping track of honeybee health and losses. Um, and typically, the acceptable winter loss reported by beekeepers historically has been around 15%. And actually, we see that beekeepers are since about 2013, maybe, getting used to having higher losses because the acceptable winter losses, reported acceptable winter losses increasing. But we're still seeing really high winter losses and total annual losses, well above what beekeepers are okay with losing. And there are several reasons for that. There are, of course, the natural parasites and diseases. There's poor management practices. So you have these large kind of monoculture commercial beekeeper operations. And I think people, if they weren't aware before, since COVID started, can look at this picture and understand how social distancing is not happening here, and how if there's virus or disease in one of these colonies, it can quickly spread to other colonies in this type of living situation. There's poor nutrition, so forage is disappearing, and in these commercial operations, a lot of times you have monoculture that honeybees are presented with for weeks at a time. And then there's the pesticide issue, not just pesticides going on to the crops that they're foraging on, but also the ones that we sometimes have to put into colonies ourselves as beekeepers to control those pests. So most of my work focuses on the natural parasites and diseases that threaten honeybee health. And I want to go into Varroa destructor and the biology of Varroa destructor a little bit more, just because I think they're such a huge problem. They are a physical burden to the bee. So I think if you had a parasite, I mean, we know mosquitoes are so tiny and a huge nuisance to us. So if you had a parasite relative to your body, the size of that Varroa is relative to the honeybee, it would be the size of like a small dinner plate or something on your body. I mean, it's a pretty big parasite to have sucking your hemolymph or your fat body. Um, it, of course, is a problem also because it vectors disease, but studies have shown that in addition to vectoring the diseases and spreading the diseases, they can amplify the number of virus particles before spreading them. So, really problematic. They can harm the honeybee in two different life stages. So they're found phoretically on the adult bees, and they're also forced, the only way they can reproduce is on honeybee larvae. And so the way that that happens, here we have this kind of the development of the honeybee in the cell. The foundress mite will enter the cell of a developing honeybee right before capping and kind of actually hide in the brood food at the base of the cell so it won't be detected. The brood eats up that food, and then the mite's released, lays one egg that develops into a male, and then all the subsequent eggs that she lays about every 30 hours develop into females. The females mate with that first male brother of theirs, and that is how you get essentially reproductively ready individuals emerging from the cell. And I believe they do have to go through a phoretic stage before they can enter another cell. But you can see that, with this one development of 1B, we can get 2 or 3 um, new foundress females. And that's why we see over the course of the summer the population of Varroa build up so quickly. Now, honeybees, like other social insects, are pretty vulnerable to pest and disease. They have, compared to other insects like Drosophila, the fly, fruit flies, they have relatively few immune genes. And then they have really high genetic relatedness. They're all sisters in the colony, and a high rate of contact. And because of that, they've actually developed several different mechanisms of social immunity. And these, going back to the fascinating bee, these are some of the things that I think are really just super interesting about honeybees, is these social immune mechanisms that they've developed. So one, of course, is grooming, and you can see here allo grooming, which is where a sister is grooming a mite off of her nest mate. You have something called brood comb fever, which works similar to the way a fever works in our body, where workers can actually raise the temperature of the brood comb and kill some pathogens by inducing a brood comb fever. Um, and then there is propolis, which I think until pretty recently, a lot of people thought was just kind of a nuisance substance that wrecks our equipment. But over the last decade or so, we've really become aware of how important propolis is to controlling pathogens in the colony. And then finally, we have hygienic behavior, and that's what I focus on with my work. And I'm going to show a video here. If you look at the middle of the screen, you'll see hygienic behavior taking place. And hygiene is the ability of those adult bees to detect, uncap, and remove unhealthy brood from the colony. So they are sacrificing a larvae, a developing pupae, but in doing so, they improve the overall health of the colony. And this behavior exists at a low level in nature. So every colony practices hygienic behavior to some degree. And because of that, it can be selectively bred for. And that's actually been done for several decades now. I would say the pioneer of this work is Marla Spivak, who developed the freeze kill brood assay and has developed the line of Minnesota hygienics based on this assay. And if you're not familiar with it, what she does is take a PVC pipe and you pour liquid nitrogen into the PVC pipe so you kill freeze kill the brood within that test area. And then you put it back into the colony for 24 hours. And colonies that can remove 95% or more of that frozen brood are considered hygienic. A non hygienic colony after 24 hours would look something like this. And so, um, studies have shown that with these hygienic lines, you can see increased mite removal, at least in some hygienic colonies. There's debate about how much that is the case, and we'll come back to that later. But I really think that in terms of sustainability, breeding of Varroa resistant colonies is one of the most, if not the most sustainable option. When we talk about pesticides, the The mites, of course, can evolve resistance, so it's kind of like a continuous arms race against those mites because we develop a new pesticide, a new miticide, and then they gain resistance. And on the other hand, we have lots of things like, drone removal or um, caging the queen, brood interruption, but those are all pretty labor intensive.
Kaira,
Yes.
could you speak to a too hygienic queen or hive where the uncapping goes beyond the beneficial level to the colony?
Sure. And yeah, I'll get to that a little bit later too.
Okay.
Um, but I think that I have seen it in some hygienic colonies where I've actually had colonies so hygienic that they are removing even healthy cells. And I think that's a problem. But I think there are ways to avoid that. So I'll come back to that, and then if you have more questions about it, let me know if I don't answer your question. So there several different hygienic breeds or Varroa resistant breeds that display elevated levels of hygiene. So a couple of examples, there's the Minnesota Hygienics. It's selected based on freeze kill brood. There's Varroa sensitive hygiene, which is originally based on mite removal, and now I think is selected also based on non-reproduction. These terms kind of get all twisted and they keep getting redefined. But then there's also right now the pollinator line coming out of the USDA that's selected based on mite non-reproduction. And so I think there's advantages and disadvantages to the selection methods for these different lines. So on the one hand, we have freeze kill brood. This is a relatively rapid assay. You pour the liquid nitrogen in, wait for it to thaw, take it out, and stick the frame back into the colony. But liquid nitrogen can be difficult to access for a lot of beekeepers. It can be difficult to work with. And with the Minnesota hygienics, I think the consensus is that we really only see mite resistance or significant reduction in varroa mites when you have those highly, highly hygienic colonies, so 95% or above. On the other hand, we have the poll line from USDA. They've shown that they can get actually really good mite control with these colonies, but it's not a selection practice that I think is practical for most people. So they're looking in up to 1,400 cells per colony and, using a microscope to look at mite reproduction and how many offspring are in each cell. And so it can just be very tedious. And so I think the problem is that with these selection methods, you tend to see a trade-off between the efficacy and the efficiency. So there's a need, I think, for an easy, rapid, and more precise selection assay. And so when I started working with honeybees, I was really interested in the communication system between the signal emitter and the signal receiver in this hygiene in this hygienic behavior. And when I started, I think a little bit was known about the signal receiver. Definitely more was known about the signal receiver. So we knew that hygienic bees had better olfactory sensitivity. They're better at smelling, better at detecting problems in the cell. We also know that a lot of hygiene is performed by specialists. So in a colony, there's actually just a few bees that are performing a lot of the hygiene. But we really didn't know a lot about the signal emitter. So what's this signal that they're detecting? So the first thing I wanted to know was, does the brood contribute to hygienic behavior? And so what I did was I took 3 different lines of honeybees and I did a cross fostering experiment. So I actually took queens and caged them on a single frame. And then I cut those frames into thirds and kind of re-grafted them together, not grafted like the normal grafting of eggs. But I would cut the three frames together and then put a frame back together that had, some VSH, some hygienic, and some unselected Italian eggs on it. And then I would stick that frame with three different types of eggs back into a new colony. When the time was right, right after capping, I would introduce mites. And then what I did is I looked at those treated cells and monitored them for hygienic behavior. And what I found was a positive correlation between the hygiene of the brood's colony of origin and the likelihood that the brood was removed. So, in other words, it didn't matter so much where I put that frame, that regrafted frame, as it did which section of brood I was looking at. So, what really mattered the most in my experiment was where the brood originated from, how hygienic that colony was that the brood originated from. And so this showed me that, wow, well, brood chemicals are probably really important to hygienic behavior, but at that point they weren't very well characterized. So we know that chemical signaling is really important for communication in many different social insects. It's important for things like defining family membership, nestmate recognition, territoriality, mating, resource quality and location. So of course that's how the ants find their way back to your picnic blanket. And then things like necrophoresis. And this last example, E.O. Wilson played a major role in explaining what was going on with necrophoresis, or the removal of dead bodies in ants. And what Wilson noticed was that an ant could be, completely dead, legs up on its back, and just was completely ignored um, for the first maybe 24, 48 hours. And then what he noticed was after some time had passed, then the nestmates would recognize that something was wrong, pick the ant up, and take it to the disposal site. And what he found through a series of experiments was that there was a chemical called oleic acid that was kind of the signal of this death, known as a death pheromone. And it turns out that this oleic acid is also found in, for example, freeze-killed brood. So those bees that are freeze-killed for hygienic behavior tests. But I think what's becoming clear is that the signal emitted by a dead brood is not the same as a signal that's emitted by a diseased or parasitized brood. So you can imagine that a dead brood might send out a much stronger signal than the signal that an unhealthy but still living parasitized brood may emit. So honeybees are the epicuticles, or the outer layer of honeybees, are dominated by hydrocarbons. So all different kinds of chemicals are found, but hydrocarbons are the most common. And this includes alkanes, alkenes, which have a double bond, and methyl alkanes. Um, and so just to show you where that is, you have the mite and the brood. On the outside is the cuticle, and the very edge outer layer of that is the epicuticle, and then you have the hydrocarbons kind of stacked up on that epicuticle. And the main reason for those hydrocarbons being there, at least initially with insects, is resistance to desiccation or drying out. It's kind of like a waxy layer that prevents them from drying out. And just like babies get cold faster than adults because they're small, they have a higher surface area to volume ratio than adults. It's the same with insects. Insects are small, they have a lot of surface area, so they're very um, susceptible to desiccation. But social insects have kind of co-opted those hydrocarbons to use for communication. And so the next thing I wanted to know in my studies was, well, do these cuticular hydrocarbons or CHCs from stressed brood trigger hygienic behavior? And if so, which ones are responsible for triggering hygiene? And so I designed another experiment. I took capped brood. Some was parasitized with mites, some not. And I extracted, basically pulled off those cuticular hydrocarbons or CHCs using a solvent. And then I ran two experiments. So in one experiment, I took just regular, healthy, unparasitized cells, and I applied to the caps, the wax caps, either extracts from the control bees that had no mite or extracts from the mite parasitized brood. And the other thing I did was I ran the samples through something called, or analyzed them using gas chromatography mass spectrometry, which basically just tells us what chemicals are present and how much of each chemical is present. So in the first case, what I found was, when I applied that mite-infested brood extract to the cell, those cells were more likely to get uncapped by adult bees compared to the ones that were treated with extract from a brood that did not have a mite on it. When I looked at the results from the gas chromatography, I see something like this. I'm sure it looks like nonsense to you. It kind of looked like that to me at first. But basically, each one of these peaks represents a different chemical. And then I can spread these peaks out, and the volume under each peak tells me how much of that chemical is present in the sample. And so what we found, to make a long story short, is 10 hydrocarbons that were elevated in unhealthy bees compared to healthy bees. And by unhealthy, I mean mite parasitized, or they had higher levels of deformed wing virus, even if a mite wasn't present. And so what I found was that, there were these 10 chemicals, but there were a couple that were really important. Z10, C33 is one that I found over and over to be elevated in unhealthy bees. So, for example, if a mite was present, we saw this chemical in greater quantities. We actually never found any chemicals that were new that weren't present also in healthy bees, but we saw just a change in the relative amount of certain chemicals. And this was a cool finding because it's actually consistent with some previous findings. For example, a paper in 2017 showed that this same chemical is elevated in ant pupae that are targeted for unpacking. So I think evolutionarily, this isn't something that has just come about with honeybees, but may go back a little bit farther evolutionarily. Um, we also found that it's a similar structure, this chemical Z10C33, to other chemicals that have been linked to hygienic behavior. So when I say similar structure, we found different chemicals all had a single double bond. The double bond was always on an even numbered carbon, and the total molecule always had an odd number of carbons. So there are just some specific details of this class of chemical that we noticed. So we've identified 4 chemicals, all with this kind of similar structure, and all of which had been linked either by us or previous work to Varroa and or hygienic behavior. And so what we think now is that we've kind of come up with a new model of hygienic communication, or um, a new idea of how it may work. And it's important for honeybees because we know that there are these specialists that perform the hygiene. They have to be able to somehow get the attention of those specialists. If they're, on a different area of the frame or on a different frame, how are you going to get them to a problem area? Well, the way you have to do that is with small volatile chemicals that kind of go out and draw them in. So they're coming into the higher concentration. um, but if all they have is these volatile chemicals that are kind of mixing all around the colony, then there would probably be a lot of errors. So a lot of opening the wrong cell. Is it over here? And so what we think is that there are also these larger chemicals that are present that are non-volatile that really help those specialists pinpoint exactly where the problem is. And this helps reduce error in terms of hygienic uncapping. So the next question I wanted to ask is whether these alkenes can be used to measure Varroa resistance. And that's where we come to this novel tool that we're developing for Varroa control. And we call it the unhealthy brood odor or UBO assay. And so the idea is we took these chemicals that we were interested in, synthesized them in the lab. So they're exactly the same as the ones that are naturally produced by the honeybee, but we can make them in the lab so we don't have to kill a lot of brood to collect them. We made a mixture and then applied that mixture to capped brood. And then after 2 hours, so it's kind of designed similar to the freeze kill brood assay, um, but instead of a 24-hour waiting period, we do a 2-hour waiting period. And then at 2 hours, what we do is check uncapping and removal. So this is an example of the test area, and I do a much smaller test area to compared to the freeze kill brood assay. We do about a quarter the number of cells, so it's about 45 cells in the test area. Um, at time 0, we spray the pheromone mixture onto the cells. And then at time 2, we take a look and see what's happened. And what we see in a hygienic colony is we get a response like this on the left. And in a non-hygienic colony, we may see a few uncapped cells, but definitely a very different response from the hygienic colonies. And so we took 72 colonies and tested them with the UBO assay and the freeze kill brood assay. And then we checked them for mite infestation. So we did powdered sugar just the first year. This is data from 3 years of um, of research, and now we have, I think, 5 sites involved. And we calculated the mite number per 100 bees. And what we saw was that both assays could actually predict mite infestation. So when we performed our UBO assay in June, and we use a threshold, kind of like the freeze kill brood assay, based on our data of 60%. So we consider a high UBO colony to one that scores 60% or above in terms of uncapping and removal on that UBO assay. And what we found is the colonies that scored high on the UBO assay had an average of 1.6 phoretic mites per 100 bees, compared to 7.2 phoretic mites in the colonies that scored low. And these are colonies for research purposes that have never been treated. So we're not knocking mites down in any of these colonies, which is why we have these high numbers. We saw a similar trend with the freeze kill brood assay, but the numbers were a little bit closer together. So the high freeze kill brood assay had an average of 3.2 phoretic mites compared to 6.8 phoretic mites in the low. Um, and I think the interesting point here is I don't know what you guys use in terms of treatment thresholds, or if you use a treatment threshold. But the latest recommendation I've found is at about 3% you should treat your colonies. And so I think it's interesting that the UBO assay can kind of distinguish the colonies that fall above and below that treatment threshold, whereas that line is a little bit less clear with the freeze kill brood assay. And I think this explains why there's variable results in freeze kill brood assay in terms of Varroa resistance. Next, we looked at mite removal. So we introduced mites to individual cells and then looked at removal of those cells 8 days after those introductions. And we found similar results in this case between the UBO assay and the freeze-killed brood assay. So in this case, both assays were able to distinguish colonies that were better at removing experimentally introduced mites from those colonies that weren't so good at removing those introduced mites. And then the last thing we looked at is overwintering success. And what I found was that the UBO assay scores were much higher for those colonies that survived overwinter than those that died overwinter. Of course, I'm talking about the assay done before they died. So we would test them in June and in August, and the August UBO assay was pretty good at predicting whether a colony would survive, and I think that's primarily related to Varroa infestation. So these colonies that are scoring high on the UBO assay have fewer mites and are more likely to survive. So I think this is important work because what we're trying to do is develop an inexpensive and beekeeper-friendly tool that can help facilitate management decisions. So for example, I think it's useful for beekeepers because they could potentially reduce their miticide use, ID susceptible colonies that they know they need to treat for Varroa, and maybe avoid using miticides in those colonies that can take care of themselves. You could also potentially compare queen breeders. So if you get queens from a couple different sources, you could test their hygienic level and know who you want to order from in the future. But I think it's also really important for breeding programs in the US. So what I'd like to do is test this in terms of an improved selection method for hygienic honeybees. And I think it's better than existing models because it uses selection based on an actual honeybee stress signal. So remember, these pheromones are pheromones that are naturally found in those unhealthy honeybee brood. And it gives us better hygiene resolution. So I think this kind of comes back to that question that was asked a little bit ago. With the freeze-kill brood assay, hygienic colonies, or those mite-resistant colonies, are really only those colonies that are scoring 95 to 100. There's no real wiggle room within that 5% to tell what's going on. But with the UBO assay, we really see pretty solid mite resistance in a larger spread of response, so 60 to about 100. And so I think that's useful because maybe we can now use this assay to distinguish, okay, well, on this end of the spectrum, maybe it's really still not quite sufficient number of infested brood that are removed. Maybe on this side, they're so overly hygienic that they're removing even the healthy non-infested brood. It's like an obsessive compulsive behavior, and then maybe in the middle is somewhere that is ideal for selective breeding of improved bees. And of course, improved management, improved hygienic selection means improved honeybee health and thus better crop and natural ecosystem pollination. So that kind of wraps up the first part of my talk. And I want to move in now to the second half, which I promise is shorter than the first half, so I shouldn't call it a half. But I kind of think of it as, you know, this first portion of my talk is, um, you know, I really had a question. I planned it out carefully. I thought about it a lot um, and looked closely, and I discovered something interesting. But what can also happen in science is that something interesting discovers you, and that's kind of how I think about this second portion of the talk. So if you remember when I kind of that first experiment I showed where I was splitting the frames for that cross fostering experiment. So I was out there, you know, kind of just learning beekeeping, and I was trying to saw these frames into thirds and had plastic foundation in there, and it was kind of a nightmare. It was like sweating and wasn't working well. And so I even at one point tried like heating up metal wires and cutting through the plastic, and I was like breathing all these fumes. Not good situation. So I ended up switching to wax foundation, which experimentally, for some reasons, is not as great because we're not always easy on our bees. We move them around a lot, and it's not as sturdy. But it was definitely a lot easier to cut those frames into thirds. But what I noticed really quickly was something like this, a pattern like this in my comb. And I'm really interested to hear from you guys, maybe in the question and answer section, how many of you guys have seen something like this. But I saw this, and clearly there's these lines of missing brood, and they line up with the metal wires. Um, so I know that honeybees are really susceptible to stress during development. So we know they're susceptible to pesticides, they're susceptible to poor nutrition, trace metals, and that's not surprising. So in a matter of just a few weeks, they gain 1,000 times their weight. They have really high metabolism, really fast growth rate. So it's not surprising that they're susceptible to stress. Well, we know that stress actually impedes population growth, and that bigger colonies are more likely to survive over winter. So my thinking was, well, if the population of the beehive is affected by these metal wires, maybe it's important to overwintering success. So I wanted to learn more about it, and I did a literature search, and was really surprised to find that I could only find one study that mentioned any effect of the wires. And what the study said was that honey that was collected from those cells right along the wires contained 4.2 times more iron. So I thought that was interesting, but nothing was out there about the brood. So I was wondering, okay, well, what is the effect of this steel wire on honeybee brood? And the frames I was using were stainless steel, according to the company I purchased them from. So I tested the effects of the steel foundation wires on the chemical content of the brood and brood removal rates. And so the first thing I did was I collected larvae that were basically found right along those wires, cells that were lined up with those wires, and then cells collected larvae from cells right next to the wires. And I sent them off somewhere else. And they looked at the chemical content of several different elements, and the only difference I found was iron, and that matched really well with the honey study. But brood along that wire contained more iron than control brood. And the next thing I did was I counted larvae along and adjacent to the wire. I returned, so I didn't remove them, I just counted how many were there, returned them to the colonies for a week, and then came back and I counted how many were still there. And what I found was that the wire was associated with a 6.4-fold increase in removal of brood. So I did this in 5 colonies, and in every case, we saw a really big difference in the number of individuals that were present along the wire and adjacent to the wire. And all that counting was assuming that we had kind of this perfect alignment of the cell to the wire. But what I noticed is that that's not always the case in reality. So sometimes the wire is offset a little bit, and what we actually see in that case is kind of almost this zigzag where you get twice the effect of the wire on brood cells. And you can kind of see that here. In some cases, you get this kind of straight line like this one on the left, at least at the bottom, and then you can see in others, and at the top here, that kind of zigzag where you get twice as much removal. And so there are about 3,300 cells on one side of a deep frame, and that means there are about 198 to 396, 200 to 400 cells overlapping the wire, depending on kind of this orientation if they're all aligned center or all aligned kind of off center. And what that amounts to is 6 to 12% of the cells overlapping the wire. And so I think this was a really interesting and important study because one, you have the reduced brood survival, obviously, but then there's also things to consider like the wasted resource. So think about all the food that went into rearing those brood, all the time and energy that it took not only to feed them, but then to remove them later. You also have to think about compromised worker health. So if they're removing these brood, a lot of times honeybees are also cannibalizing the brood as they remove them. And studies have shown that iron containing nanoparticles may be important to navigation. So this is a study showing iron containing nanoparticles in the abdomen of a honeybee. And so if they're cannibalizing these iron rich brood, that could have an impact on their navigation ability to navigate. So, just to summarize what I've told you overall, I've told you that honeybees are important. Hopefully, you knew that already. But they're important for crops, they're important for natural ecosystems. I've told you that honeybee health is declining and showed you that Varroa is a key factor in that decline. That unhealthy brood can trigger hygienic behavior via chemical signaling. That the UBO or unhealthy brood odor assay can predict mite removal and overwintering. And I think from that we can say that the UBO assay can predict colony varroa resistance. And I've showed you that steel wire stabilizers can be harmful to honeybees. So I still have a lot of questions about the wire stabilizers. This study, I haven't gotten more funding to continue it. So I still have questions about, for example, all of the frames I used were brand new. Does this, once you go through a couple brood cycles, does this dissipate? How important is that if we're being told to replace our frames every couple years? So I still have a lot of questions with respect to that study. With respect to the UBO assay work, we have received a ton of interest in this assay, most of it for selective breeding purposes. And we have lots of big plans for this summer. So I have plans here in North Carolina, but we're also doing studies at the Baton Rouge, Louisiana USDA apiaries in France, Israel, Canada, and the Netherlands all have plans for studies this summer. I'd like to look more at predictive ability of the assay in terms of varroa sensitive hygiene specifically and disease loads, productivity and overwintering success. So is there a difference in honey production of colonies that score high on the UBO assay? And then because it's such a quick assay, there's some studies that show you can get 15, 20 minutes of reduced olfactory ability, reduced ability of the workers to smell when you're using the smoker. So I want to know if the smoker affects the honeybee's ability to smell, maybe that could have a significant impact on the assay. So there's more things that we need to know about the assay and how it works. But we're working on finding those out, and we do have plans, hopes to bring the assay to market. So this is done all through my university, but they have several patents out now on the assay and are working towards to bring it into the commercial market. And then one thing I just wanted to mention that I'm working on now, I'm really excited to do a study this summer on a priming effect. So in 2019 and 2020, we did some experiments with our assay where we tested the same colony twice within the same week, about 3 days apart. And in both cases, we found that the colonies responded more hygienically to the second assay than the first. And so my thinking is that there could be some kind of priming effect where you kind of get these pheromones into their antennae, they get exposed to them and they think, yikes, there's a big Varroa problem, or there's a big need for hygienic behavior, and they either recruit more hygienic workers or they up their those specialists up their amount of hygienic activity. So I'm really excited to look at that more closely this summer, because I think that has opportunity to be and not just kind of a management tool and a selective breeding tool, but a treatment for Varroa. So if you know you have a Varroa problem and you can put pheromones into your colony to kind of stimulate hygienic behavior, that could be a really neat and sustainable treatment for Varroa. So with that, I will stop. Hopefully everybody's still awake over there. I have done this not by myself, but with lots of collaborators and important funders like Project APISM, my university, the USDA, NC Biotech, and have had a lot of great mentors along the way.
Yeah, this is Phil. One of the questions that I put in the chat is this very sensitive, subtle scent that these bees are picking up. How much are we disrupting that with mite treatments or even our smoke, right? I mean, we go in there with a smoker and blow their noses out for a couple of days, right? Are we completely disrupting them with some of that or with some of the mite treatments?
I don't know about the mite treatments. I don't know their effect on olfaction, but that's an interesting question. The smoke, what I understand with the smoke is that it has a temporary effect. So I think it's maybe 20 or 30 minutes. So I doubt, I think that could have a big impact on a 2-hour assay, but I doubt it has a big impact on overall hygiene in the colony. But I don't know about the Varroa treatments. That's a good question.
I know it bothers my nose. It's got to bother theirs.
Yeah, I tend to avoid smoker use for my own comfort as much as I can. Not that stings are comfortable if you got a hot one.
Yep. So one of the things you also mentioned was the specialized bees, and that's in last month's Bee Culture magazine. There's a great article about 3 bees I have known, and which is a fantastic article by Gene Robinson, talking about some of the bees. He had one who was just a caretaker. It lived its entire life as an undertaker, right? And he followed that bee, and all it did was empty out hives. It never turned into a forager. It lived its entire life, and so they had some other specific characteristics on some of these bees that he followed in there. So on this Varroa sensitive hygiene, I'm wondering how many specialists do you think there might be? Could there be like a dozen and they spend their life there and they teach a couple dozen more and that's about it? Or are there thousands?
Also, great question. I don't know how many there are, and I think that's something we still have to untangle. Is a colony that's hygienic, does it have more specialists, or does it have the same 20, 30 specialists, but they're just better at performing the job? And that's something I don't think we know yet. But would definitely be interesting. That's one question I have that I'm hoping to tackle a little bit this summer. So many projects.
Duncan asks about the horizontal wires, which I presume would behave similar to the vertical wires. And Phil suggests nylon fishing line.
Yep. So I haven't tested the horizontal wires, but I think they would, as long as they're kind of crossing the brood in the same way, I think they would have a similar impact. I have a lot more questions. I I really want to know more about how long that effect lasts. Because if it is just the first couple of brood cycles, I think it's less important than if it's something that keeps happening over and over and over. But I don't see any reason why horizontal wires would be different. I have had people suggest fishing wire in the past. I've never tried it myself, but I think it's a great alternative to these wires. The ones I was using were supposedly stainless steel. I don't know if that wasn't true because the stainless steel isn't supposed to leach as much. So either that wasn't accurate according to the company, or it just still had a negative effect.
I'm assuming, I've never heard of stainless steel vertical. It's called crimped wire, and no manufacturer I know of produces it with stainless steel. And horizontal wires, generally a talented frame builder, the wires don't really, you embed them in a way that they're not really impacting the frames very much. And they do diminish after the first round usually. And it's just physically the wire being in the space where the queen would lay the egg. And it's an interference. So there's so few people that use wired and wax foundation anymore. It really is not a big deal. It's kind of a, but in general, it goes away very quickly after first use.
Yeah, when I did this study in 2015, that was the #1 product sold by the company that I purchased the frames from. So I think the amount of use has decreased, but in North Carolina, people still are using this primarily, the wired wax foundation.
That's really interesting to know. There, you have old timers. That's good.
We do. Certainly.
But I've noticed on some hives I'm taking care of that given the space and the option, because they're wires that are horizontal, they will build the comb around the wire if they can, not necessarily, embed it in their wax foundation. They'll like go around the outside or go in the inside. Not always a straight game.
Interesting.
So part of the construction of using wire and wax is you normally embed the wire so it's in the wax. If it's ever if the wire's ever out of the wax, then it's not constructed properly.
No, these are foundationless frames that.
Oh, got it.
Yeah.
Yeah, and in this case, the wire was embedded in the wax, but there was still, I guess, enough contact. I don't think the wire was actually physically touching the brood, but was leaching through the wax.
I've used that style of vertical crimped wire, and in 25 years, I've never had bees not lay in the vertical wire space. What you showed is the first time I've ever seen that, and I've been using it for 25 years.
That's interesting. So they did actually lay in it. They just removed them. So they made it to about 5th larval instar. I don't think most of them did not get capped, but they made it to about 5th larval instar before being removed.
And now it'd be really cool if we can get mites to taste like iron, and then they would remove them all.
Right? Just got to feed the mites iron somehow. Has anybody else seen that pattern in their brood?
I have not.
When I've seen new wax foundation with wires, I think the wax is shiny, but if you ever pull a wire out of old foundation, it looks like it's pretty corroded. It's kind of got a patina. So I wonder if that's from the chemical action of the various acids in the honey on the wire.
Mm-hmm.
I've never seen that lack of, that kind of hygienic behavior along a wire. That was a first for me.
Interesting. Yeah, I was definitely surprised by it.
20 years, I've never seen it.
You guys
I'll be
must
looking
use a
now.
different wire over there.
So this is the only kind of foundation I use, is the horizontal 2 wires in both mediums and deeps, and the vertical wires supplied by the foundation. I do embed the wire with electricity to, get it embedded in the wax, and I'll pay way more attention to it, but it has not caught my attention that there's any indication of a pattern there.
Yeah, I think it's something that if you're keeping an eye on your frames, you would certainly notice if it's there.
I used to use wax foundation that I got from Brushy Mountain probably 15 years ago, but I haven't been using it for many years.
I use it today.
Did you notice
The vertical ones? The vertical ones, Hal?
Yes, both. I buy the vertical ones. I just got 25 pounds of it, and I put in 2 horizontals in the middle 2 holes in case I ever use the frame for extraction. Every one. The only thing I use plastic is if indeed I'm selling bees and I do a frame exchange and I get some plastic in. That's I kept bees for a lot of years before this, and that's what I did then. And some people it's hard to get a dog to learn new tricks.
So Kaira, did you see that same pattern in your control group, or only in the hygienic bees?
So that was actually control bees. That was before I was working with hygienic lines. So that was all unselected Italians that were doing that level of hygienic removal. That wasn't even hygienic bees.
One would think that if we're going to see that, and we're not doing the treatment, I'm sorry, not doing the study, that every once in a while you might see that in bees that have higher level of hygienic behavior. But thus far, maybe my girls are lazy. I haven't talked to them lately.
I think it's probably the type of wire, honestly. And maybe just different companies are using a different type of wire, and there's more and less inert wire types. I was certainly surprised by it, and I've had when I tell people about this study, I typically have a handful of people say, "Oh, I've seen this in my colonies." So it's not everybody, but people see it, and I don't know the difference. I don't know why some people see it and some people don't.
Yeah, well, the wire that's in the wires. Excuse me, the wire that's in the manufactured foundations clearly isn't stainless, but the wire that you buy from bee supplies, to do the horizontal wiring is definitely a stainless, stainless wire. So, you're using both kinds.
Actually, Hal, the Kelly was my favorite wire, and it was a tinned, I don't know what the base steel is, but it wasn't stainless. If you're going to do stainless, you have to specify and get it from a special source, and it's pretty expensive.
Really?
So it's just that, yeah. And a lot of the normal wax that you get from Man Lake or any of the normal sources, I found it was a little more like wet spaghetti. And the Kelly, I hope they don't change it now. There's purchased by Man Lake, but the Kelly wire was actually had a final tin layer on it. And the tensile strength was quite a bit more, which I was happy about. So, but I've never used stainless steel, and I've never been able to, I could always purchase it specifically, but it was much more expensive and from special sources.
Yeah, I was always under the impression that it was stainless, and it always kind of teed me off because if you overheated it slightly, it snapped. So
right.
I'll try the Kelly stuff. That sounds interesting.
Yeah, we'll all have to ask and see, because Man Lake purchased Kelly.
Right.
I don't know about their sourcing anymore.
Yeah.
So Kyra, if you want some of that old, wax foundation with the vertical wires that I bought many years ago, I still have some I could send you if you want to know
Thank
what
you.
the wire was.
Yeah, I mean, it was advertised as stainless steel. I'm curious. And it was actually from Brushy Mountain, which I think is where you said you got yours.
Yeah. So,
and that's since closed down.
Exactly. I mean, I might even say on the box where it came from, just to make sure I was right about that. But if you wanted to test what kind of wire it was, I'll just let you know that if you wanted it, I could send you some.
Okay, thanks. Yeah, that would definitely be interesting to kind of delve back into that and see what it was and what's being offered now, and if there's a difference.
Mm-hmm.
The fast, easy test is just rip it out if you have one. Just rip it out and wipe it down and set it out in the out on the picnic table, and you'll know in a week or two if it rusts, whether it was stainless or not.
Right.
It's also non-magnetic.
There we go.
The stainless is non-magnetic?
Yeah, most of it, depending on the type.
Yeah.
Kaira, I had a question. Did you explore at all the issue of bees using propolis as part of the capping? Because you had mentioned that earlier on in your presentation that that's one of the adjuncts to their immune system.
Mm-hmm. Yeah, I haven't studied propolis. I know that it's important. I don't know about its use in capping, but I know that it's important and volatile. So it doesn't actually have to come in contact with a pathogen to kill it directly, but it actually releases volatiles in the colony space, and those volatiles have antimicrobial activity. I am related to that, really interested in cell cap architecture. So one thing I'm interested in looking at is, are there differences in, for example, the porosity of hygienic bee cell caps versus non-hygienic cell caps? Because I think that um, there are pores in the cell cap. That's how the bees, the larvae, breathe and get oxygen. So it has to be porous. But I think there could be differences in how that cap, the architecture of that cap, allows those volatile hygiene signals to cross through. And so that's another thing I'm interested in.
What about the content of the capping I understand that some pollen is mixed with the wax in order to cap it and make it somewhat permeable so that there is some gas exchange, oxygen exchange. Mm-hmm. So do you know whether certain types of pollen are contributing to, or are you going to be looking at that, contributing to the architecture of the cappings?
I don't know. That's a great question. Yeah, that's something I haven't gotten into yet, but that's on my to-do list. So that's one of the questions that I'd really like to look further into, is how I hadn't thought about the different types of pollen. That would be interesting to look at, because there are, different shapes, different sizes. But maybe just the content, how much pollen is present, how much of that bee bread is there, how, what's the weight difference between the caps of different colonies. And you can see, visually they look very different from one colony to another. But I don't think people have paid much attention to the architecture, and so I'd like to look.
I saw a study from Europe where they compared honeybee health in bees kept in Langstroth, Warre, and Layens hives, and there was a difference. I was wondering, when you're doing your studies, do you only use Langstroth, or do you try your studies in some of the other hive types?
Yeah, we only use Langstroth in our bee yard. But what was the difference that they saw?
There were differences in the number of larvae, and the amount of honey produced, and the general overall health of the hive.
Interesting. No, we currently only use Langstroth. We have so many experimental questions we have to answer that we try to limit the other variables as much as possible. So at least for now, we stick with Langstroth. But another interesting question. It's the thing. The more we learn, the more questions we have. So that's why I love science.
This is a little off topic, but I was interested at the beginning that when you were talking about the difference in feeding different bees and how queens are fed better food, resulting in the queen. And then you were talking about queen longevity. Do you think that any of the queen longevity issues have to do with quality of the food? Or what do you think about the change in longevity of queens?
Yeah, certainly. I think they could. I think queen health in general has been significantly reduced. I think it's probably like honeybee health in general, that it's kind of a multifactorial issue. So I think miticides are not good for queens. I think you know, forage quality has been reduced. I think that even semen quality has been reduced, and that could impact queen lifespan. Maybe not her natural lifespan on her own, but if she has reduced semen quality in terms of what she can get on her mating flights, then if she doesn't get as much, or if the quality isn't as good, then the colony is going to requeen sooner if they sense a failure. And so, yeah, my guess would be it's really multifactorial.
So on the semen, do you think that it's a question of a problem in the mating areas? I mean, do you think that the queens are getting less drones that they're mating with, or what would be semen quality?
So it's anecdotal, but I heard from Sue Kobe told me that she's just seen a difference in the way the semen looks over the past decade or two. So it's just changed in the way it looks, and I would guess that's related to pesticides and specifically miticides, the introduction of miticides. But I don't know. I haven't looked at that. That would be that's all anecdotal and a guess on my part. But we certainly don't see those, I mean, I've never had a queen that lived 5 years. I don't know about you guys.
I've had queens that lived in the past easily 3 years, and some of them 4. I don't know if I've ever had a 5-year queen, but I've just in the last few years seen an unbelievable decrease in the lifespan of a queen.
Oh, really?
To the point where the queens, sometimes they're superseding twice a year.
Wow. That's interesting that you've seen it so recently, just in the last.
It's very recent, and I don't see why pesticides or miticides would have changed. I mean, I'm not sure I understand what that could be. Forage quality around here is really good, I think.
Mm-hmm. Yeah,
So
I don't think in the last 2 or 3 years miticide use. Well, I don't know in your area, but I don't think it's changed that drastically.
If anything, I would have thought it might get better, but I don't know.
Well, I also think that the level of nutrition in what they're foraging is decreasing.
Why? I mean, we have actually an area that I live in, it's just filled with forage for bees that's very diverse. We have trees and all kinds of things. What has gotten worse? To me, it's a
the level of nutrition in it.
Why would that change?
global warming.
Higher CO2 in the environment, so a lot of the pollen counts and the nutrition in the pollens have gone down. Nutritionally, food is a lot less nutritious than it used to be.
Yeah.
So it's just tied with a higher carbon in the atmosphere has reduced the food nutrition and the pollen nutrition pretty much across the board. So it's yeah, you may have the same cherry tree on the block, but it just ain't putting out what it used to, unfortunately.
You think the fires could be involved with that? I don't know how much worse they've been in the last couple of years compared to the last 10 years.
Oh, terrible.
Oh, they've been terrible. Last year was
Terrible.
awful.
Yeah, no, we've had terrible, terrible fires, and the smoke is very widespread, way beyond where the fires actually are.
So I wonder if that could, if it's been a big change in the last couple of years, that might be a hypothesis to start from, that the fires have had some impact.
I've got mostly the same era and the same trees as everybody else, but I've seen a lot of green dot queens in the last month. So
sorry, a lot of what?
Green dots, which means they're
dots?
green.
Oh, green.
Yeah, they're 3 years old.
Yeah, so I've got a lot of those. So I'm not finding that same.
Nikki, do you grow your own? Do you raise and grow your own queens, or do you buy them from the producers?
No,
they're mine.
No,
they're mine.
I got you.
How they're my queens, and they're from like I've been trying to breed them over years, so they're from the same lines. But I can't control the drones, and so I don't know that those lines still have the same characteristics as they did 10 years ago.
Yep, and you live in an area where inbreeding isn't an issue.
No, I don't think inbreeding would be an issue here. So I don't know. I'm finding it really mysterious. It's quite a challenge.
Beekeeping is local.
Beekeeping is local.
And challenging.
But, I also live in an unusual neighborhood that has a great diversity in tree species and a lot of plants in people's yards. It's a suburban area, and but just it's a very tree-laden neighborhood, and planted to have many diverse trees. So, I mean, other people might have less diversity in their forage. I have very high diversity in forage, and maybe Phil's hypothesis and your suggestion, Kaira, to look at the smoke or to look at the change in carbon. I don't know.
Yeah, or inbreeding also could be a possibility.
It just doesn't seem likely that we have inbreeding here. Mm-hmm. But it would be fun to, have some study that looked at the mating areas.
Yeah, or if you brought in some new genetics somehow, and
we have so many, feral hives, beekeepers. They're not all doing the same thing.
Mm-hmm.
My couple of lines are not affecting the inbreeding potential. Yeah, I don't know. It's a mystery.
So many questions.
Yeah.
Kaira, have your queens come from diverse sources, or do you mostly breed your own?
So I don't breed my own. I've used every year I get new queens, so I try not to use the same queens in subsequent years, just so I get new data. And I have used primarily unselected Italians from standard breeders here. They're mostly from South Carolina, Georgia area. And then I've used the Minnesota Hygienics produced by Amy Weeks and Jeff Hull, the Pole line out of the USDA, the VSH line out of John Harbaugh's apiaries in the USDA. This year I have some Russians I'll work with as well. So I've pulled from a couple different sources every year.
And the assay pretty much works with all of them?
Yep. So I also have, I guess the only breeding I've done is I do like to make splits and requeen from like an Italian maternal line in an apiary where I have mostly hygienic bees and see kind of what those hybrids do. So, you know, not really breeding, but I like to have some hybrids in my studies as well. And we just see, you know, really a pretty straightforward correlation between like the scores on the UBO assay and how resistant they are to Varroa in terms of those phoretic mite counts, regardless of the line. So if I have a really hygienic unselected colony, they'll score high on the UBO assay and they'll have a low Varroa mite load.
Given how difficult it is to control the drones in breeding programs, you have said that you were hopeful that this assay would be helpful for breeding projects, and I'm certainly very interested in that myself. Do you think that, or have you, the people that you've talked to who are doing breeding, are they all doing artificial insemination, or, you know, are there ways to have some control over the drones without having huge areas where you have few bees?
Yeah, great question. I think most people are concentrating genes using artificial insemination that I've talked to that are like larger scale breeders. But I think that it's definitely possible, especially if we can get a lot of people to start using the assay and breeding based on it, then you can improve the drones kind of in your area. But I think I've also heard a lot of people say, well, I would test not just my, you know, my maternal lines, but I would test all my colonies and then choose some to be drone producers, put a bunch of drone comb in those colonies, and some to be the grafting colonies where I collect the eggs for the queen cells. So I think you can kind of, you know, if you have, even if you're in a crowded area, you can kind of saturate to some extent the area with drones. My understanding is that queens fly farther to mate than drones, so that I think that's kind of a system to limit the inbreeding of drones from mating with a queen from their own colony. So you might be able to play around, especially if you know where those drone congregation areas are. You could potentially play around with putting your drone source colonies near those congregation areas, and you know, kind of tilt it your way spatially. But I think a lot of people are using insemination to some degree.
Well, that's an interesting idea about identifying where the queens are going to mate and then increasing the drones in those areas. That's a really interesting idea. Do you know anybody who has been trying to saturate their areas? It's an approach that I have thought of trying to do. It seems to require, you know, really getting people to, you know, kind of all coordinate, which is more difficult. But do you know of any examples of people doing that?
Yeah, I mean, I've talked to people who definitely, you know, if they know a good set of colonies that they want drones from, we'll put a bunch of drone comb in. I don't know about moving them to. I haven't heard of people specifically moving them to congregation areas, but I don't see why it wouldn't work. You know, especially if the drones aren't flying as far as the queens, you put the drone source colonies closer to where you know those mating areas are. But I think, you know, it would take some coordination, cooperation with your neighbors. But if you guys have a, you know, a tight beekeeping community in your area, and you can get people on board and say, look, let's all improve the drone quality in this area and see if we can get better hygiene. I think that's definitely doable. It just depends on your beekeeping community and how cooperative, I guess, your neighbors are.
It's a really interesting idea.
Randy Oliver is probably the biggest example of someone who creates a very large drone area. He's completely surrounds his mating apiaries, and then he gives queen cells to all of the beekeepers in his immediate vicinity, so that they're all running his genetics.
Yeah, that's a great point.
I've heard of other people doing that, but what Kaira just said was that queens are flying further than the drones, so saturating around your apiary.
Well, he's got a lot. He's got probably 30 apiaries in the Grass Valley area, so he's able to saturate a huge area.
Yeah, and that's a great point. I've heard of other breeders who really just try to like give away their queens and drones, which, you know, I think isn't an option for everybody. But that's definitely a good way to saturate the area with what you want genetically.
I've heard of that too, but it's been in places that are not suburban, and people have a little bit more control over their, over their area. And so it's definitely not suburban, and Randy Oliver's not exactly suburban either.
Well, I know people in Europe are doing some of that, where they're really kind of giving away their genetic lines to kind of increase the genetic stock.
I've heard a number of breeders talk about doing that. But like I said, that's fine if you have a fairly rural area, but it's more difficult when.
Well, I think in Europe, it's actually pretty dense in terms of colony. So the guy I was talking to was in the Netherlands, so was doing that.
In Germany, the old school way they did queen mating was to send them to, you put them in special boxes and send them to an island where it was very controlled. And I think for the same reason, I don't like to breed my bees in the urban area. I have an isolated mating yard, and I don't know. I think Hal's probably kind of isolated for most people too. So I think as a choice, we try to breed our queens away from everybody else so that we don't have mutts and a mixed, questionable source of donation.
Well, one of the things that's come up in our Alameda County beekeeping ordinance standard debate with the sheriff's department is we have somebody on our committee who's trail, he's a East Bay Regional Park supervisor, and it occurred to us that the parks, which is the largest landowner here in the Bay Area, allows cattle to graze on their park lands, and there's some relationship between the cattle owners and the parks. And could the beekeepers not strike a deal with the parks to create our own breeding yard for bees?
Interesting. Yeah, because then you can know that you're the only hives in the area.
It wouldn't be the only hives in the area, but it would certainly help to have more control over the drones. And,
yeah, you'd have the feral stock. So
right. Yeah.
Well,
you'd have the feral stock,
it's not even the feral stock. I mean, some of the larger beekeepers in our area thought that they were going into the park and doing whatever, but they found there were other beekeepers there. So it's not like there's no other beekeepers there or there's no feral stock. You have some control. It's better, I think, than in the urban and suburban areas.
You might be able to know better what is there and have somewhat of an island effect. The other method I wanted to mention is there's I can't remember what it's called, but they have this interesting method that they developed, I think, in Greece, where so most mating flights, as I understand it, happen in the morning to midday. And so a lot of those the drones that are flying are going to those congregation areas earlier in the day. And in Greece, they developed a system. It had a really funny name. I think it was like the Virgin Train, something like that. But they kept all their colonies with virgin queens in like cold storage, and then they would pull them out in the evening, and they would only release those virgins in the evening, and they would also, during like that same 2-hour period, release a bunch of drones from the stock that they wanted, the genetic stock that they wanted. And so the idea was to, by timing it when most drones are not out flying in the evening, they were force mating those virgin queens with the drones that they wanted. So that you could also play around with the timing, if you can control release of your drones and release of your virgin queens. But if you Google like virgin, it's a little bit nerve-wracking to Google it because I had to do that too. But like virgin queen train, something like that, in Greece, I think I saw like a little 5 or 10 minute video that explains how it works, and it was pretty interesting.
You know, in California, we have some areas that are really desolate in terms of the beekeeping because of the forage that's there. We have the buckeye tree that is very toxic to bees. And it's almost impossible to get a colony through the season with them. And I've had pretty good luck feeling like I'm getting a very similar take on mating there. I only use it in the early spring, March and April, until the trees start to bloom, and then I pull them out. But the hard part about it is you just don't know what saturation is and how many colonies you have to bring out, and especially early in the spring, even as we really don't have winter here and we have drones flying in February. But there are areas, if you hunt them around, Sue, I really think that's a great idea to approach the parks, because there's so many areas that they have that are insular.
Yeah, I talked with Chris Rocha, the trail superintendent, on this the other night, and the challenge that they're finding out is unfortunately, some local beekeepers are advertising that they're raising bees on the East Bay Regional Park land, and of course the park district is a little up in arms that their name is getting slapped on someone else's honey. Not the fact that they're not getting a cut, but there's liability and all the rest of that stuff. Mm-hmm. So they're probably going to be, coming down a little bit on that. So if you are keeping bees on East Bay Regional Park land, you'd probably be advised that you want to watch out for your hives, because they're doing a roundup on all of that right now. But like Sue said, right, that does pose an opportunity for us to propose this with them and having a relationship so that we could make some really great drone congregation areas, or work with the park district the same way the cattle ranchers do to, set that up. I mean, they've got parkland, and as Jim has proven, and a lot of other people who have done it in the back hills, it's only good in the wintertime when, in the spring, when you've got good forage. By the time June runs around, there's no point in keeping a bee out there anyway.
Right. It's, dirt city. So it's really a short window.
Yeah. But it's a interesting prospect.
Yeah,
definitely. We all just need islands to be able to take our bees to. I did find the link to that that video clip about the timing of drone and virgin release.
Yeah,
if you guys are interested, I posted it. Yeah, I posted it there in the chat.
Okay, I gotta go, but thank you very much for your talk. That was wonderful.
Absolutely. Are there any other questions?
I just wanted to mention that in terms of beekeeping in agricultural areas that are being overtaken, we have a huge solar plant going in North Livermore. And the agricultural plan specifically designates that the usage of the land for beekeeping will be a big part of the agricultural plan. So, and I have a good friend who's part on the committee for that who mentioned it to me. And of course, it'd be nice to be involved. But that's also a situation where, in this particular case, you wouldn't be insular, but it would be easy to, or it may not be easy, but it would certainly be possible to populate the drone property, population with your drones more than others.
Yep.
Well, we're looking forward to hearing when you have this assay available, or if you're wanting to work with people before it becomes available. So that was very interesting and we appreciate it.
Well, thanks. Hopefully it'll be out there soon. There's certainly a lot of interest, and I think from this summer we'll learn a lot more about the capabilities. But I'll stay in touch.
It was a great lecture and so well presented.
Excellent. Well, if any other questions arise, please don't hesitate. Jerry has my email, and I'm always happy to talk bees, talk hygiene. It's what I do all day, and I love to talk about it. So,
is this talk recorded?
We are recording it. Yes.
Great.
Yeah, Kaira, great work.
Thank you for sharing.
Really good work that you're doing. I love it.
Excellent. Thank you guys so much, and I love the discussion afterwards. It's refreshing to have such active members talking and asking and discussing. It's great.
And on behalf of the club, thank you for letting us keep you up late.